Hi, everybody, and welcome to Neuroscientists Talk Shop, UTSA's neuroscience research podcast. Today is February 27th, 2025, and we're talking with Xue Han, who is a professor of biomedical engineering at Boston University. Xue's lab is working on optical methods of recording and modifying neural activity using genetically expressed proteins. She invents the molecules that we all use for optogenetic stimulation and for optical recordings, and she validates them so the rest of us can use them with confidence and know that they're going to work and know what their properties are. That's the bioengineering part of it. And then she also uses them in her own neuroscience research program to answer basic questions about how the brain works. And today, special interest for us will be methods for measuring membrane potential using voltage-sensitive fluorescent proteins. Hi, Xue. Thanks for joining us.
Hi, Charlie. It's great to be here.
And I'm your host, Charlie Wilson. So for podcast listeners interested in a little bit more of the story of fluorescence recording of neuronal activity, I would suggest a couple of our previous episodes, specifically episode 246, which was William Ross Retrospective. It was posted on June 2022, and Bill Ross was a real pioneer in calcium imaging, and it's super interesting conversation with him about the history of it. But also, this conversation took place in 2007, and Bill was predicting how calcium imaging was going to pan out and what we were going to gain from it. And he mentioned its strengths and weaknesses, and everything he said turned out to be right. He is so prescient about what was going to happen with calcium imaging. So I think that's a really good thing to listen to. And then also episode 234 with Surgeon Antic, which was from December 2021. And he talked about voltage imaging, which is our same topic today, what it can teach us about neurons. And he also filled in some of the history of it that we probably won't bother with too much today because it was before. He was mostly talking about what happened before the genetically expressed indicators. So anyway, so go back and see those. So anyway, Xue, let's talk about voltage imaging. First, like these genetically encoded indicators, how are they discovered and how are they perfected? And when you want to make a new one that's better, what do you do? How do you go about making a better one?
Yeah, so when we talk about these genetically encoded voltage sensors, they need a few properties. First of all, they need to be on the membrane so they can sense the membrane voltage. And when we think about the neuron or the cell or whatever, excitable cells, and the space that can accommodate these proteins are limited. It's different from the cytosol where you can feel a lot of proteins. So keep that in mind when we have a small number of proteins that needs to be on the membrane or very, very close to the membrane, either inserted or next to the membrane. And that's one criterion, they sense the voltage. And the second thing is they need to give off a fluorescence. So that fluorescence needs to be voltage dependent. And so those are the criterion, and they need to be really bright because we want to image very fast. If we do a kilohertz, that's 1 millisecond per frame. So we need to get enough number of photons from a very small number of proteins that gave off these photons. That means these sensors need to give a lot of photons, be very sensitive in a very short amount of time. So that makes it a little challenging. And so starting off from the voltage sensitive domains, there are a few classes of them. One of the popular ones is actually the rhodopsin molecules. They have these charge domains in the membrane, and they can sense voltage changes across the membrane. So they have been used recently in some of the newest protein-based or genetically encoded voltage sensors. So they sense the membrane voltage change, and then they give off fluorescence. So the voltage sensor that we have been using or developing is called SOMA-Archon. It's a single compartment. It sends its voltage, and then it changes its conformation, and it gives off a near-infrared fluorescence. It's all in the single molecule.
And this is its native fluorescence.
It's native fluorescence.
You didn't have to modify it to get it to do that.
It's native. We modified it slightly by mutating the amino acid. It's binding to the retinol, which is vitamin A, essentially, and it's a cofactor. So it's the rhodopsin, like in the eye, but these are the microbial rhodopsins. It's a native fluorescence. It's enhanced so that its fluorescent change in response to voltage is optimized for recording. So it's a single protein, very tiny. So that's what this class of this called Archon, or SOMA-Archon, or derivatives of this class of molecules.
So when you're gonna enhance it, are you looking at the sequence of it and saying, "Oh, I'll bet if I change that thing to a lysine, that residual lysine, that would do it," or do you just start sort of in an exhaustive way making changes?
So in the beginning, people try to target mutagenesis. Based on the crystal structure, we go to change some amino acid, and we have some ideas that if you change this amino acid, for example, that's the one that interacting with the cofactor retinol, that would enhance the fluorescence. And actually, one class of it is archaeorhodopsin base. It's actually a light-activated proton pump. So the first thing we need to do is to kill its proton pumping ability, because otherwise it's measuring voltage and passing the
changing the voltage at the same time.
Exactly. So that has to be done, and then we need to get rid of that and then enhance this so we can go to that pockets, but that doesn't do all the job. That's why Archon was a randomized mutagenesis, and just randomly you screen millions of potential mutations and came out and see which one works better. So you need to have a screening platform and find these molecules. So that's how Archon was evolved from the original quasar protein, but they all evolved from archaeorhodopsins. So just lots of mutagenesis, and you get lucky ones, it works. And then we get a few potential targets, and you have to figure out which one actually works in neurons, and which one works in the brain. And there's a lot of variations and a lot of luck involved. Yeah.
And they have to, is it hard to get them expressed at the level that you need? Because you must need to load the membrane with as much of that stuff as you can get.
Exactly. And sometimes you load it too much and the cells don't like it, they die, or they don't do anything, they become unhealthy. So there's kind of a balance that we need to have sufficient number, and they need to be on the membrane. If we have a lot of them in the ER, in the Golgi, it doesn't do anything. They actually contribute to the noise. And so in the final kind of the molecule we use, we have a lot of tags to help them go from ER, Golgi, and actually targets and concentrates to the plasma membrane.
So this is making use of decades of learning about how proteins get targeted to different places on the cell.
Absolutely. And also, taking advantage of all these optogenetic molecules, because they don't always go to the membrane, like the halorhodopsins. They are famous for sticking into somewhere in the ER, in the Golgi. So we have to help them to get to the plasma membrane. And so for these voltage sensors, they need to be on the plasma membrane. That's where we are measuring. And that's another advantage of the genetically encoded ones, is that we can target them to the plasma membrane. Whereas if you have a chemical, it's hard to target. They could stick to any membrane. And then the thing that's not on the plasma membrane, that contributes to the noise. Yeah.
And then it matters where on the cell it is. I know that when the voltage indicators were loaded into cells just by diffusion, and they would load all the cells, and what you got was this sea of changes of fluorescence that you think, that must mean something, but I have no idea what it means because it's every cell in the whole place is contributing.
Right. That's another advantage that people like genetically encoded ones. Then you can target specific cells, and there's all these, cell-specific targeting technique with the Korean enzyme, with these rodent models. And you can target this cell specifically into, cholinergic cells in the striatum, for example. And so we can target, so only those cells will have these voltage sensors, and the rest of the cells will not. Then we can get very good signal, not only from these cells, but improve the signal to noise, because the rest of it has no noise. Because the thing you don't want, that's your noise, right? And so the rest of the tissue is dark. So that's helpful with improving signal to noise as well. Yeah.
So why do we want this? Why do we need voltage? What's wrong with what we've already got? People say this all the time because they go to a lot of trouble to learn some method, and then along comes somebody with this other method. They go, why do I have to learn that? So there are microelectrodes that give us really good temporal resolution, calcium indicators that allow us to look at more than one cell at a time. What are we missing in all of that?
You are the expert on intracellular recordings. We want to know what the membrane voltage is, not only just the spike, because the membrane voltage decides when the spike is generated. You have input, but that input may or may not drive a spike, depends on the membrane voltage. And so for all these years, decades of research, we can do this really well in in vitro system. We can patch neuron, we can get the membrane voltage, but it's really hard to do in the brain during behavior. And so that's why we settle on these microelectrodes. We can record spikes. So we listen to the output, we listen to the spikes. And in vitro system, we can get the rich dynamics. We can even figure out which ion channels and what is the kinetics. We can explain a lot of it. But then once you put everything together, the inputs of the neuron in the brain is so dynamic. We don't really know where it's from, and it's probably really hard to figure it out. But then, how do we make sense of things? So if we can get this subthreshold information, which somehow is kind of resemble what we saw or we understand in a principal way in a piece of brain slices, but add all the extra noise that the neuron would receive in the brain, and maybe that's when we can start to understand what the spike trains that we get actually represent. So if you take spike trains from one neuron, many neurons, can we get to the system level understanding of a brain circuit? Some people believe we can, right? And other people think, well, we are missing a lot of information. So exactly what we are missing, I guess that's ultimately we have to figure it out. Like, for example, I think if we look at synaptic potentiation learning, reinforcement learning, that's some information we can't easily get from the spike trains. Can the EPSPs or the IPSPs or the synaptic potential changes? And I think inhibition is another thing that's hard to get. So I think these synaptic changes would be really interesting to look at if we have the technique. But what are we going to discover, right? I'd love to hear what you think in the end. What is the fundamental insight that we will actually uncover that we cannot get from just the spectrum alone?
Well, a long time ago, I used to record in vivo in anesthetized animals using intracellular recording. It wasn't a thing that everybody did, but there was a reasonable community of people doing that. And of course, a lot of people recorded extracellularly and just knew about spike times. And I was often asked, why are you going to all this trouble to get the intracellular recording when I can tell you when the cell fires? And of course, the answer is, oh yeah, the action potential tells you the output of that neuron, but the membrane potential tells you the input of that neuron. So the neuron is actually getting input from maybe 10,000 different other cells in the brain, and the next neuron is going to fire differently because it gets input from 10,000 different cells in the brain. And you're looking at just those two neurons firing, you just know about two neurons. But if you record intracellularly from one neuron, you know about a huge ensemble of neurons that are all functionally related to each other and are all controlling that neuron, and you can see what they're doing together. as are they just making chaos, or are they collaborating to create some pattern in this neuron? So for just looking at sort of spontaneous ongoing activity, the intracellular recording is telling you about the input. And you can, it's the only way to get that, because the only other way to get that would be to somehow identify the 10,000 neurons that project to that cell, and then record from those 10,000 neurons specifically right now. And of course, you're not going to be able to do that. So that was one thing. And the other thing that I think most people who are using intracellular recording, when I was doing it in the 80s and 90s in vivo, were doing was using it to find out who is connected to whom in the brain. So they would stimulate one group of neurons and they would record from another. And just as you said, if neuron A inhibits neuron B, it's not going to show up in the spike train unless neuron B is firing really fast to begin with, and so you can make it slow down. And not all the cells you're interested in are firing really fast to begin with, and we'll reveal that. And also the inputs are graded, and there's a whole lot of interesting stuff that happens before the cell spikes that determines what's going on. And so those are the two reasons. I think it's almost forgotten because there's been very little opportunity. There's still people who record intracellularly in vivo, even in behaving animals, but it's considered heroic. And it's a really a very serious thing for the most serious people to do. It's, I think, hard to get new students interested in such a hard job.
It's too hard. Yeah. But to add to that, another thing is that we do local field potential recordings. And also that's being done a lot in humans. And we don't, local field potential is just anything next to the electrode. I think even though we don't have absolute evidence, but I think a lot of the thinking is that it really captures the synaptic input way more than the spiking.
Yeah.
Because spiking is so brief, and it's the amount of current going through each spike is actually very tiny comparing to the slow but massive amount of synaptic current. So I think thinking about even just from an oscillation point of view, especially with this increasing amount or number of patients that have these intracranial electrodes, they get the local field potential, or they call them IEEG, whatever the name they call them. They got these frequency domain sort of analyses. And then you try to understand what they are. And I think those are probably best thought of as synaptic inputs. And that's probably captured through, you do intracellular recording. So
synaptic input, though, just smeared across the entire population of neurons.
That's right.
So if this neuron is getting one kind and this neuron is getting another, the field potential will have both.
That's right.
And you won't know that those two are different signals.
Yeah, but in pathology, still, most of the biomarkers, even in human patients, they are looking at those signals.
Yeah.
And there are some that's really well established, especially in epilepsy, for example. So it's really interesting to look, how does these epilepsy biomarkers related to these intracellular signaling of the individual cells around the region where the seizure starts. So that's, for example, one really clear kind of a translational perspective why we also need to do these intracellular recordings, because these recordings can bridge what we know about neurons versus what we see in the pathological brain in humans. Yeah, and that's something's not really spiking. Really.
And it's not just one neuron at a time. Another big drawback of the microelectrode intracellular recording is that the number of neurons you can record from is so limited at any one time. There's some people that record from 3 or 4, but that is also very heroic and isn't done very much.
That's right.
So in something like epilepsy, that is it is a circuit pathology,
Absolutely.
Then you can't really capture it. One neuron at a time, you need to see what's happening across a group of neurons. So let me just point out another thing I think is bad about intracellular recording with microelectrodes. Although I think it's wonderful, I love doing it, but I always wanted to know what the dendrites are doing. And again, you can try to impale dendrites and record from them, but it's so hard, and it usually isn't done. But with optical methods, the dendrites are just there for the taking.
Yes and no. So I think for in vivo recording, so far, most of the voltage sensors that's so sort of developed is membrane is somewhat targeted so that we can label a few neurons, and when we do in vivo imaging, we can find them. If we label all the dendrites, it becomes a little harder.
You won't know which dendrite goes with
because
which
it's
neuron.
all over the place, and then you lose that contrast. But I think it's possible if we are sparsely labeled some neuron, then we can see the dendrites nicely. So with the existing sensors, even though they are targeted to the soma, but they are not completely to the soma, we can label some of the proximal dendrites 300-400 microns from the soma. We can see them clearly. We can see these signals. I have not yet figured out what is the use of them, but I think in the future, something along those lines. It could really, like starting from the proximal dendrites, for example. And then we can develop some better sensors. And there are now better sensors that we are actually hoping develop. With those, we could do sparse labeling and track dendrites, and perhaps, I'm hoping, that we will be able to record along the dendritic tree in certain experimental conditions. That would be so exciting. We'll be opening up so many new frontiers. Yeah.
There's been this great theme. It's really more conjecture that was shared by lots of people and gets repeated over and over again. But there's definitely evidence for it. The dendrites have their own active properties, and they can do things. And with voltage imaging surgeon, when he was here, showed us these beautiful dendritic spikes in basilar dendrites of pyramidal cells and he could use voltage imaging to see where they were happening in the dendrites. And that is a real clear demonstration that kind of thing can happen. But once we start thinking about that, we start imagining the dendrites performing logical operations, right? So this dendrite gets one input, this dendrite gets another, and then they interact with each other to produce some result. It's kind of like what we thought neurons do, only now it's happening just between two dendrites, and maybe the cell has 25 dendrites. And so each neuron can become a much more complicated logical machine. And there's been a lot of speculation about what that could mean for neural circuits. And this is going to, don't you think? This is going to let us actually look for that eventually.
Right? Like, how do we even start imagining the computation and dendrites? Once we start seeing, then we can start to think about what they are doing. Like right now, I think you can do this probably rather even now, you can do this in a brain slice. You can see the dendrites. It's much easier, right? But once in vivo, once we start tracking them, then we can start to understand really what is the rule for dendritic computation. Like right now, I don't even know. I don't even know where to start to think about this. But I believe that. I think there's the dendritic compartment the dendrites are so long. It's got to do a lot of computation along the dendritic tree. Yeah.
And they're branched, and different branches get different inputs. So there's automatically a kind of logical operation going on between the branches, even if they don't have, sort of dendritic action potentials.
Yeah.
And if they do have dendritic action potentials, then they become even more.
Yeah. I think we also know the different inputs from different circuits could synapse on different parts of the dendrites. They all laid out in these beautiful ways. And so if we can start recording those, I think that would really allow us to imagine what is this computation like. Yeah.
So one of the things that seems to me could be happening, but I don't see it happening very much, is using optical recording for membrane potential and then stimulating pathways. I think once we start working with freely moving animals, there's a lot of changes in input coming to a neuron all the time. We get this very complicated pattern, never really repeats. The neuron is getting a different input every minute as the animal's doing whatever they do. And so I think that kind of causes people to not want to, stimulate the prefrontal cortex and see what am I going to get in some spiny neuron in the striatum, because the next time I do it, that something else will be happening. But I think if things are organized the way we imagine them, it would still be very clear, if some cortical area went just to the distal dendrites or went just to one half of the dendritic tree or something like that, we would see that stuff. It's another one of the things you think that your anatomical studies have told you, but in fact the anatomical studies don't tell you that. Because in most anatomical studies we end up averaging over many observations, and some of them are over here and some of them are there, and we just think, the pathway does both things. But it may not be doing this both things all the time, one thing on one cell and one on another. So I don't think we know that much about exactly where inputs end in the dendritic tree.
Yeah, would it be great to do such experiments?
Yeah.
And some of the voltage sensors, like the Soma Archon, is actually near infrared. And so it can be combined with blue light optogenetics. So if we put the optogenetic sensors in the input, then just illuminate at the same site, give some blue light to drive those inputs, and then simultaneously we can image the membrane voltage of the postsynaptic neurons, then we can probably start to answer some of these questions. I think these experiments are totally feasible with the existing tools, and I think just need to start exploring these different concepts and experiments. Yeah, so I think it's great. It's just a lot to do, and I think it's good to get this into neuroscientists' hands and then to get this set up so people can do these experiments. Yeah.
So one drawback is getting imaging in the part of the brain that you want to get an image of, right? Some places are easy.
Right.
Some places are especially difficult.
That's right.
And should we all just move to the work on the cortex where it's right on the surface or on the cerebellum?
Well, that's part of the issue, and we struggle with as well. Like, we record from the striatum, we record from the hippocampus, but we cannot reach. So we take out whatever is on top, and now we hope that doesn't change too much of whatever we're studying, but it does. So that comes down to the interpretation of results, given the controls, and this is what we see. But then you can always ask the question, what if the cortex is intact? Can you still say what you say? Can you derive the same conclusion? So I think there's always this idea, like when we try to observe something, we are changing the system. So for voltage imaging, we need super high speed, like a kilohertz. 2-photon line scan is just slow. There is some 2-photon that can go to kilohertz. Those are very difficult to work with. You have very specialized instrument. So most of us are in the wide field condition. Means we can image for up to about 100, 150 microns from whatever the glass surface. So that really doesn't get us very far. Even in the cortex, that's probably layer 2, barely 3. So layer 2, 3, that's the minimum, that's the maximum. And then if we want to go to the hippocampus or the striatum, essentially the overlaying cortex needs to be taken out. So there are people, and we are also part of that crowd, is trying to develop techniques with fiber bundles, green lens, these really kind of fiber optic methods, so we can drive that deeper in the brain. There are a different set of challenges, like once we image this fast, is there autofluorescence from the fiber technology, the cladding of the fiber, and so on. So these are the things many people are working on, and also driving this field in the voltage imaging field specifically, high speed, this color wavelength, and how do we get images? So not only just the fiber photogrammetry, you get one channel. Can we do all the neurons deep in the brain without too much damage? So that's another thing. I hope that soon the field will figure out what to do about that.
If I want to image the hippocampus or the striatum, which are both directly under the cortex, how big of a piece of cortex do I normally have to? I'm picturing the objective on my microscope, and it's like this big around. It's like the size of a quarter.
So, yeah, there's also these prism that is curved. So there are various ways, and if we just don't care, just take out something so we can look. I think 2 to 3 millimeter, that's a lot of tissue to take out. And then this comes down to the covered glass where you're going to get these tiny, very specialized stuff. And then we make a cannula, we put it in there. So I think somewhere like around 1 to 2 millimeter or 3 millimeter, that's probably the ballpark that you can image through. And there's this long working distance lens with very high numerical aperture that can do it. So that's not too big of a problem. But, a lot of things need to be aligned to get a good imaging window. So I think still there's a lot of tissue damage, even 1 to 2 or 3 millimeter. That's a big piece of cortex we're taking.
It's not just for one day. You make that hole in the cortex.
Absolutely.
You put something in there, and then do you leave it in?
Yeah, it stays there. It can be like a year, 2 years. And then, that's the surprising part. The mice don't seem to have much problem. This comes down to, like how much of the brain can compensate for these injuries. And in mice, we don't see much of, they don't have any kind of behavioral damage. They learn, they walk fine. They don't even seem to have that problem even the day after the surgery. Yeah.
This is an old observation that's been made many times that the cortex can be removed from an animal and you can barely see the difference. You have to use very sophisticated measurements to see the behavioral deficit. So maybe we're taking off a piece of cortex that isn't absolutely critical for at least for the things you're asking the animal to do.
Yeah, but still, then we worry, right? We're taking a lot of it out.
Yeah.
Yeah, what if that changes everything? So I think that's
definitely... Study striatum. They know most of the input to the striatum comes from the cortex. So you're taking off a big piece of the input to the part of the brain you're studying when you do that.
Absolutely. Yeah, that's definitely a concern, a limitation of the technique. I think multiphoton is still hard. It's just to get to the really fast kilohertz speed for voltage imaging. People can do it, but it's really limited, the field of view. A lot of them just studying like the membrane, so you scan the membrane. But then once you get to the behavior, there's always micromotion. Blood flows, there's micromotion on the scale of microns, submicrons, and if you are in a very precise scanning field, that could be out. So that just requires a lot more surgical precision and so on. So I think ultimately this is a debate. What is the better one? Wide field imaging or 2 photon? They each have the drawbacks. Wide field certainly has more sensitive to out of focus light and so on.
So there must be a lot of image processing required from these images because the brain is bouncing around, the blood pulsations are happening in intracellular recording normally in vivo. You worry about just the pulse from the blood flow. It can cause you to lose the cell. It means they're moving. So I guess you have to correct all of that. How do you do that?
Yeah, so we typically get the images and we look at them, and then usually in a wide field mode, the cells have, let's say, some motion. We can do image correction, just realign the images back together. And we typically take a look first, and if the image motion is too big, we just can't use that preparation. And sometimes it just happens, for whatever reason, the imaging through the view is not stable. Because we have a covered glass, so this prep is actually good. It serves as an anchor. It anchors the tissue around it, so it moves less, I think. So we could have very, very stable windows when we get lucky. So there are very little motion, and then you can see the motion, you can see the outline of the neuron. And so usually, when we see that, we reject the whole recording. And sometimes, unfortunately, if you want to study locomotion, when the animal moves, there's more image motion. So that requires better surgical techniques. So sometimes we can use it when the animal's not moving. And once they start to move, we just cut. So these are all the...
So if you have 3 or 4 cells that are all moving together, they're not moving independently of each other. Is that right? Isn't it like image stabilization on a camera or something like that just corrects when the whole thing moves?
Actually, it's interesting. You would think that, such small field of view, right, there would be moving together, but sometimes they don't. We definitely see that the rigid image correction algorithm doesn't quite work, so we have to do some more sophisticated ones to do local corrections. But usually, yes, they all moving, but then the amount of movement that we need to correct for is not always the same across just the same field of view. So image correction is definitely the first step, and then I do think that's something definitely need to be factored in. We always look, say, this is the image, I'm shifting it back, and the amount of correction, we need to plot it and make sure the amount of correction is not contributing to whatever my interpretation is. Yeah, so that's something we always check, and I think that's something people should be thinking about. Yeah, it's definitely something, the mouse does something unless they're absolutely still. Otherwise, they could have motion. And if we're studying breathing, heartbeat, this could all create micromotion. So these all needs to be checked. That's my personal take on this image correction issue.
So it's technically still very challenging for people to do this. You have to learn about the molecules, you have to learn about the microscopes, you have to have the surgical techniques, you have to have the software to do the imaging.
That's right. And also the data size is tremendous. It's kilohertz, and we have lots and lots of data. So we actually have the supercomputer cluster that does a lot of the work, because each data set could be tens of gigabytes. And now if we record very long recording, sometimes it's like a terabytes of one day recording. We can't run it on our local machine. It just runs out of memory. So there's a lot of issues, and sometimes we can't continuously record because it fills up the memory, and we have to stream directly to the computer memory. If you do a solid state disk, there's a delay of the transfer of the data. We learned that, and then they cause the problem with the sampling rate. So we actually use memory, directly storing memory.
So you have to be able to hold the whole movie and
then we stop the recording 1 minute on or 2 minutes off, and then offload that to the solid state drive, and the next recording starts. So that's another thing that we have to deal with. Yeah.
Of course, neurophysiologists have had to do this for a long time, but the volume of your data is much greater. On the other hand, the size of the disks around now are much larger. I think this is a problem really sounded familiar from the way things were.
But now it's just, it's just this data set is even bigger. Yeah.
Well, I'm looking forward to being able to do that. I just think it ought to be, is it going to become, of course the computer problems get solved. One thing we've learned is the computer problems get solved. Basically, disks get faster and memories get bigger and computers. I don't know about cameras. Cameras are getting better all the time. Can we expect what is a typical camera that you would use for 1 kilohertz recording? What does it cost?
Actually, the students were just asking me about that. What does it cost to do these experiments? The ones we use are Hamamatsu cameras and the CMOS cameras. Well, I don't remember on top of my head. I would say 20,000 is about the ballpark. Around 30-ish thousand. The camera is probably the most expensive piece. The objective lens, if you want a long working distance, that could be a few thousand. And then the microscope is just...
so it's mostly know-how. That's the obstacle, not money.
Yeah,
Isn't that nice? That's the way it ought to be.
Right. So I think it's the whole setup is definitely very reasonable considering the operation cost of neuroscience experiments. Is 2 photons is extremely expensive. It's prohibitive for most labs, but for these is pretty simple. I think it's way magnitudes of lower cost. So I think it's.
I think we can expect to see this just proliferate. Everybody's gonna, once they understand what they get from it, it should be what everybody wants to do. At least it's what I want to
do. Yeah, I think with the voltage sensor getting better and better, and I know there are upcoming ones that are even better than what we have, and I think soon enough people will be able to do this in their experiments in their labs, and all those things will be worked out. It's just like in the early stage of optogenetics, the first few years, it is this problem, that problem. And over time, it got resolved. And now I see this with the G-Camp recordings. And now most lab can do it. And I think in a few years, most labs should be able to do voltage imaging.
Great. Well, thanks a lot for joining us. And it's been great talking to you about this. And very encouraging.
It's great to be here.
So this has been Neuroscientists Talk Shop.